Hello, everyone. Hello. Welcome to yet another installment of the Minds, Technology, and Society Seminar with funding from the Glushko and Samuelson Foundation. Today, our speaker is Dr. Heather Bell, Broccard-Bell,
Either one.
who is currently a postdoc in biological sciences at the University of California, San Diego, working on honeybee communication, which we'll talk about today. Dr. Bell got her PhD, Broccard-Bell got her PhD in neuroscience, behavior, and evolution from the University of Lethbridge in Canada, working with Dr. Sergio Pellis, and has a really interesting wide variety of interests related to communication, evolution, robotics, information, etc. Yeah, really excited she could come and join us today. So, without further ado.
Hi, everybody. Thank you for that lovely introduction, Paul, and thank you for the invitation to come speak. It's an honor. Okay, so today I am going to take you on a long and winding journey through the adventures of my research at UCSD. And I'm going to tell you the story of why we think that noise might be an adaptive feature in biological systems. Maybe, maybe not. We're working on it. So to start out with, what is noise? So noise, as we define it, with respect to communication at least, is anything that interferes with the transmission, reception, or interpretation of a signal. So what is a signal? A signal is, broadly, without going into the deep philosophy of it, something that conveys information, something that goes from one organism to another, changing something about the organism that receives it. And with respect to noise and how it's typically conceptualized, it's generally been seen as a bad thing because it does interfere with this signal transmission. I have to also mention that noise is not maybe just restricted to this sense as it applies to communication, but things like broad behavioral variability might also be considered noise. And as I said, traditionally we have viewed these things as being bad, or at the very least something that systems have adapted to, so something that they have learned to cope with. And this is because all of the systems that we study are biological, and biology is founded in evolution. So evolution is the thing that creates these systems, and evolution, as we know, can only work with the stuff that's there. It tinkers sort of haphazardly putting stuff together. And so I like to use the old creationist argument against evolution. And creationists always say, well, you know, evolution, if it's true, it's like the equivalent of a tornado blowing through a scrapyard and making a fully formed, they usually use 747, but here I'm going to use a car because it's easier to find a picture of something that's closer to something that evolution would create. And of course we know that that's not really a correct way to view what happens in evolution. It's not one tornado blowing through a scrapyard and coming up with a fully formed functional thing all in one shot. Evolution would be like a million or a billion tornadoes blowing through a scrapyard, and every time it blows through, it adds like one little piece that's like vaguely shaped, and it doesn't even have to be shaped for the airplane or for the car. It just has to be functional in a limited sense for something at that particular time. So it could have been a random mutation that happened to be adaptive for one reason, and then later on evolution builds structure on top of that, and you end up with stuff that looks like this. And this is a car, of course, but it's functional as a car, but it's also very noisy. It's certainly not an optimal car, and evolution doesn't build optimal things. So the car works, and importantly, it works better than things that aren't cars, or things that don't have these particular features. And so you just deal with it. So maybe noise is just because evolution builds these kinds of structures that work well enough, or at least they work well enough relative to their competitors. But lately that idea has been changing, and actually to some extent we've always known that that's not 100% true, because as we know, a fundamental element of evolutionary change is that it rests on variability. So if you want to include variability in this idea of noise, then we know that it's always been a feature. And we know from looking at gene networks where we see noise, so genes will get expressed more or less at specific times. And this, of course, has phenotypic consequences downstream. And as far as we understand it, this is the result of stochasticity, so some noise in the system. And this produces, of course, phenotypic variation, and this is where selection actually acts. So there is no evolution if there's no variability. There's no evolution without some kind of noise somewhere. So in some way, we've known that noise, as I said, is not just a bug, but it can be a feature. But in more specific systems, we have recently found evidence that noise can in fact be adaptive. This is sort of a broad diagram. This would be something that would be familiar to most of you here, of decision making. So a decision making model where we have heterogeneous elements. So this would be something like a neural network model, where there are excitatory elements, inhibitory elements, and there's some kind of balance in between them. If, for instance, we're creating like a winner-take-all network, we know that if you don't add noise to something like that, where whether or not a decision gets made or what the decision is rests on the state of the network at any given time. If you don't add a little bit of noise to that, the system runs the risk of becoming stuck in certain states, and then a decision can't be made. And then it also seems that in network models, if you don't have noise, they just don't work as well as ones that do. So this is a theoretical application. But then we know about this phenomenon. Who's heard of stochastic resonance? A few. So this is stochastic resonance, and this was a thing that was first discovered in physics, actually in earth sciences, but has since been discovered in the auditory system. We know that there are populations of cells that do this kind of thing. This is where you have a detection threshold. The system has a detection threshold. So in the auditory system, this is the stretch receptors attached to the hair cells that detect vibration, which gets sent to your brain as sound information, or it gets interpreted as your brain as such. But in order to be detected, you have to exceed some threshold. And if you have a weak signal coming in, so that's what the solid line is, you can see that if there isn't this noise, which is added afterwards, on top of it, it never exceeds the detection threshold. But we know in the auditory system, or at least in some auditory systems, in frogs I believe this is the most studied one, there are populations of cells that actually inject random inputs into the receptors, into the mechanoreceptors. And every once in a while, especially at places where the signal peaks, it occasionally gets randomly bumped above the detection threshold. This is the phenomenon of stochastic resonance. So stochastic resonance facilitates the detection of a weak signal, which seems kind of counterintuitive. You're adding noise in order to detect a signal. And as I said, this has been now discovered in a variety of different phenomena, and it's actually been implemented in engineering applications in order to boost weak signals. So noise can be good. So what does this have to do with bees? All right. So I have to back up for a minute and explain the kinds of behaviors that I study. So this is the famous Waggle Dance. I'm going to try to show you a video. If it doesn't work out, I'm not going to spend too much time trying. But I do have a video here in the background. I might, yeah, okay. I'm not going to be able to get to it. So.
Do you want 1 second of help?
Yeah, sure. This video is really cool.
Yeah, I want to see a Waggle Dance.
Let me just describe it, though. I had it open on the desktop.
Go on the desktop.
But it's also there, too. If you just scroll down and then. If you scroll down there to the bottom, it's called Waggle.
On the desktop?
Yeah. No, in the,
Right in your file.
yeah. Yeah, no.
Waggle.
Yep. All right. So, this is a Waggle Dance. So this is a returning forager. All of the members of the honeybee colony that do useful things at work are females. The males' only purpose is just to mate. That's all that they do, and they just hang out. This is a female forager. She's returned. You can tell that she's a forager. She's got some pollen on her legs here. This is inside the hive. Normally it's dark. Here we've got it open, so it's a little bit unnatural. This is a vertical surface. So this is how they normally are inside of the hive. And she's performing this waggle dance, and you can see that there are other members of the colony that are following along and watching her perform this. The waggle dance, people have noticed it, hundreds to thousands of years ago, because it's this very obvious repetitive behavior that happens in the colony. It seems like it's doing something, but nobody really knew what it was until von Frisch decoded it in, he started in the 40s, but he won a Nobel Prize for it in the 70s. And what it is it's a way for foragers who have encountered a good food source to convey that information to their hive mates so that they can now find this food source, and it encodes distance and direction. So as she walks in a line, she shakes her butt. This is called the waggle. The amount of time that she spends waggling her butt corresponds to the amount of distance to the resource. About 1 second is equal to about 1 kilometer. And the direction that she's walking in relative to straight up and down. Straight up would mean when you exit the hive, fly directly toward the sun. Straight down means fly directly away from the sun, and anything in between, that bee there was waggling, I think, at about a 90-degree angle to the right. So that means when you leave the colony, go to a 90-degree angle to the right. One neat fact about this is that if you take a forager and you put her in a box, so you've sent her to a food source that's really nice, maybe you've boosted the sucrose concentration or something, and so she's motivated to dance, and incidentally, bees all have like little individual thresholds, so you send 2 bees to the same resource, one will dance and be really excited, and the other one will just be like, and won't dance. So you take one that's motivated and you put her in a box, and you don't let her dance, and then you let her loose in the colony like 2 hours later, and she will change the angle that she walks in because the sun has moved in the sky, so she can compensate. Some people don't believe that result, by the way. I think it's a neat story, and there is some experimental evidence that it's the case. There are papers. Okay, so back to, so this is a positive feedback signal. So the longer she dances for, and that's correlated with the quality of the food source, the more observers can watch her dance, and the more they get recruited to that good food source. And this is a way, of course, for the colony to direct its resources efficiently. Now, most people have heard of the waggle dance, but not everyone has heard of the corresponding stop signal. The stop signal is now an explicitly inhibitory signal. So this happens when a forager goes out, she's watched a waggle dance, she's excited, she goes to the food source, and now maybe there's a spider on it, and spiders prey on bees, or the food source has become really crowded, so it's not very efficient to continue foraging it. In both of those instances, what it's going to do is reduce the efficiency of foraging at that site. So what she does is she'll come back and she'll locate a waggle dancer who is advertising that site. She doesn't do that by watching them because that takes too long. She does that by actually by identifying them by smell. So each particular foraging patch has a different odor associated with it. So she knows what the site smells like. She can identify the waggle dancer. And she will walk up to the waggle dancer. She'll vibrate her flight muscles, which produces a little 340 hertz pulse of sound. Actually, bees don't pay attention to the sound. They pay attention to the vibration on their legs. And she'll headbutt her. So that's what this is happening here. This is a stop signaler and a waggle dancer. I have a video of that, too, to show you. How did I get there, Paul?
That little smiley face icon in the lower left.
This one? Oh, yes. Okay, so we've got a waggle dancer here. Waggle dancer. Someone's going to come in and see that headbutt there? And then the waggle dancer just froze for a moment afterwards. And what happens is it seems to interrupt the waggle dancer, and it reduces the probability that she will keep dancing. They don't automatically stop dancing and leave the dance floor, but it makes it more likely that that's going to be the case. So what that is now is an active negative feedback signal. So it's always been a puzzle, though, like why do they need this active negative feedback signal, this explicit negative feedback signal? We only know of one other instance in arthropods. One species of ants uses a stop signal, kind of. The rest of signaling in arthropods, as far as we know, relies on passive negative feedback, so ant pheromone trails that evaporate when the food source becomes less good. Same deal with honeybees. You know, if the food source isn't very good, nobody's going to dance for it. So you might wonder, like, why do they need this active negative feedback signal? And I think that some of the work that we're doing might go toward answering that question. But the answer has always been, incidentally, this signal was decoded by my supervisor. So this is why I work on the stop signal. So his answer has always been that, well, they want to reduce foraging efficiency. Well, okay, but like that's always the case. So why doesn't everybody have these kinds of signals? And we'll get there. Those are the only movement it has. Okay. It should be mentioned that stop signals are what we call low T signals. Low T means low tightness of coupling, which means that a behavioral response is loosely coupled with the production of the signal. Who can tell me what's another example of a low T signal in something that you might study? What about input from neurons? So any particular input from any neuron onto any other input is unlikely to produce an action potential or to inhibit the firing of an action potential in the postsynaptic cell. But collectively, they have a behavioral response. And the same thing is true with the stop signal. The more of them that get produced, the more likely it is that that waggle dance will be suppressed. Yeah?
The waggle dance is not low T though, right?
No, the waggle dance isn't low T. The stop signal is low T. Yeah. But one thing that we don't know, and actually I'm going to take a little brief foray here, small diversion. One of the questions that we're asking is really, can you no, you can't really see that at all. These are some nice, some nice pyramidal neurons there. Can we actually equate honeybee behavior with neurons? That's really what we're trying to do. We're trying to build a model of how complex systems are organized, and we're trying to use honeybees as a model. Is that legitimate to do that? Because there are clearly some differences between those two things. So even if it's the case that, yeah, stop signals are inhibitory and they're low T, same deal with neurons, but you might argue that neurons' excitatory signals are also low T, so maybe it's not the same. And then, I mean, take into account the fact that honeybees can move around, and so their network shifts all the time, whereas neurons not so much. And then, of course, people have argued, well, you know, bees are way more complicated than a neuron. They can do way more things. A neuron can fire an action potential or not. But we know that that's not really true, right? Neurons are not that simple, especially with the discovery of things like backpropagation. You know, it's not this binary digital system that we thought that it was either. So maybe, maybe not. But this comes now back into noise, and why I told you about noise at the beginning. So it turns out that we don't just see stop signaling when the bees have encountered something bad. We see stop signaling happen even when there doesn't seem to be anything bad. It certainly is possible that what's going on is we just don't understand what bees conceive to be bad. So maybe we're just missing something, because of course we're always, whenever we study behavior, we're imposing our own ideas onto it. But maybe it's something else. We also see stop signaling happening that isn't directed at waggle dancers. that might not be surprising either. So my supervisor just published a paper last year showing that in a related species, not Apis mellifera, which is the species that I study, which is the regular domestic honeybee, in a species that is more common in Asia called Apis cerana, it's closely related, they have stop signaling also, but they actually produce different stop signals depending on the predator that they encounter. So a big predator produces a different frequency, actually, is what they use in their stop signal. So maybe we just don't understand all of the functions of stop signaling in honeybees. Or maybe, and this is what we think, maybe this is actually noise. This is actually signaling noise. And of course, there is precedent to this kind of thing in many socially communicating, I guess also communication is social, in many socially living species, so such as ground squirrels, for instance. This is the classic one, where they have alarm calling. We often see the case where, you know, there's an eagle flying above, somebody makes an alarm call, everybody else runs underground, yay, everybody wins. But sometimes there's nothing, and someone's just like, eagle, and then everyone runs underground, but it's a false alarm. And people have struggled to explain why that exists. So we think that that's the same kind of thing that's happening here. And we actually think that this noise might be adaptive, that not only is it noise, and not only have the bees learned to deal with it, but they've actually co-opted it for something useful. And we call this idea the beneficial noise hypothesis. And I'm going to tell you what we think is going on and give you some indication of how we've worked to try to test it. So first of all, to give you some idea of what we think it's actually doing within the colony. So we basically think it does two things. The first one is that it reduces false positives. I'm going to get into the details of why we think that in a second. But we also think that it increases foraging efficiency under natural dynamic conditions. So it allows the colony to adjust its strategy, essentially. And actually, just a comment on that. Really, the unit of study here is not individual bees, it's colonies. So I'm sure that some of you are aware of the idea of the superorganism. And this is, you know, the collective activity of the colony is what we're actually interested in here. But the collective activity of the colony has to be driven by, or probably, so the thinking goes, has to be driven by local interactions and not necessarily from top-down control. Yeah, so how does the beneficial noise hypothesis thing work? This is our assumptions. We assume, just like neurons, that bees cannot tell the difference between a stop signal that is real and one that is noisy. And every observation that we've made indicates that this is in fact the case. And in fact, we have co-opted this to do our testing. So, number two, the more stop signals that a bee experiences, the more habituated to individual signals that a bee should become. And again, we do have some experimental observations to suggest that this is the case. So in the context of building the technology that we needed to test this hypothesis later on, we looked at response thresholds in individual bees to varying frequencies. And we did see that as we played back frequencies to them over and over again, they just would respond less. We think that the more habituated the bees are, the less responsive they're going to be, of course. That's what habituation means. And the less responsive individual bees are, the more the colony on a whole is going to be resistant to any signal that's coming in, which means that any signal needs to be stronger in order to actually make it through that background level of noise. I actually just want to mention quickly that there are two cases that I can think of anyway offhand where this might be might be useful. Number one, when the colony is big. When the colony is big, there should be lots of background noise. Actually, I'm going to say that for a second because I think that this is useful to explain first. So why should it be the case that it works like that, and how does it actually confer an adaptive advantage? We think that if the benefit that can be gained from foraging at this inefficient site, so where there's a predator or where there is overcrowding, outweighs the costs of that inefficiency, that the system should converge toward the slow, passive adaptive strategy, or the passive inhibitory strategy, which is just that, you know, the problem will take care of itself because as the resource gets worse, nobody's going to dance for it. But stock signaling is fast, but it's kind of costly and it's not as accurate. The slow way is always fairly accurate. Eventually, nobody will dance for that resource. Again, the problem takes care of itself, but it's slower. But if it's the case that that relationship is opposite, so the benefits associated with any continued foraging at this bad site are outweighed by the costs of the inefficiencies, then the system should switch toward an active inhibitory strategy to curtail that right away. So where I was going before is there are really two scenarios where this is the case. Number one, if the colony is big, you would expect them to adopt the above strategy because it's not as important to them. And in particular, if there's lots of food and it's distributed in a wide area. They have lots of bees to spare, lots of foragers to send out foraging. It doesn't really matter, it's not a matter of life and death for them to forage a little bit inefficiently. But when the colony is small, then you would expect them to be more receptive to any incoming signal, and so they should maybe switch to this active inhibitory strategy. And the other instance where this should be the case is at certain times of the year, there isn't very much danger in the environment. And so when there isn't very much danger in the environment, the background level of noise, which is probably correlated with colony size, is roughly the same, but the real signal coming in is low. So the signal plus the noise is fairly low. They don't get super habituated, so they can pay attention when there is one dangerous location and curtail foraging to that one dangerous location. But at other times of the year, there is lots of danger and it's spread throughout the environment. When that's the case, the amount of signal that's coming in ramps up. Remember, they can't tell the difference between signal and noise. So when that is true, then you would expect to see a higher degree of habituation, which means that the resource has to be extra dangerous in order to curtail foraging at it, because you can't stop foraging everywhere. So if everything is a little bit dangerous. So if this is the case, it does go some way to explain why we would have an active inhibitory signal, providing it's not too difficult or costly to get there, evolutionarily speaking. So there are some specific predictions that come out of this that we can actually test. So number one, if we reduce signal or noise within a colony, we should expect that the colony will become increasingly sensitive to any stop signaling. And of course, the opposite should also be true. So if we increase the amount of noise or signal, then the colony should be relatively non-reactive when we introduce stop signals. So then the question is, how do we actually do that? And then a side question is, how long should we expect that adaptation should take? So if we're adjusting the levels of noise or the level of signal, how long do we need to wait in order for the colony to have habituated to it or not? Yep.
Just real quick, are there multiple waggle dances going on at the
Yes.
same time?
Yeah. Yeah, so It's not unusual for our, I'm going to show you the picture of the setup in a second, on our observation colonies to see like 4 or 5 waggle dances happening at a time.
Do they represent different areas?
Usually, yeah.
Okay.
Yeah. Sometimes they're dancing for the same. It depends on where the food is. If there's stuff that's relatively close by, then typically they're all going to be going to the same spot. But when it's like now, it's kind of at the end of the season, stuff is getting hard to find. They're probably dancing for multiple different spots that are far away. So incidentally, we still don't know the answer to that second question. How long should we have to wait for that to happen? But I think maybe we have found a solution so that we don't have to know that. Because, again, don't necessarily know. So this is an observation colony. For the time being, we'll just ignore this thing. It will come into play later. But basically, whereas in a normal honeybee colony, everyone's seen those boxes, especially in the almond fields. In those boxes, you have these things. These are frames. And normally they're stacked like this. But in an observation colony, we stack them vertically so that we can actually open it up and see what's happening in there. Normally, when I'm not working with these guys, there is a sheet of insulation, and then there's kind of a window, and then there's a door, so it's dark, because that's normally, they live in cavities, they aren't used to the light. When we're studying them, so this is actually inside of a trailer on the UCSD campus. We have an apiary on campus, and we have 4 observation colonies inside of the trailer. So we can actually control conditions in there. I can sit in there with my bee suit and black out all the windows, and arthropods aren't particularly sensitive to red light. At least bees aren't particularly sensitive to red light, so we can use red light. You know, people say that they can't see it, but every time that a bee flies somewhere, she flies like immediately to the red light, so I think that they can see something. Anyway, it's supposed to be less disruptive, so that's how we normally work with them. Open them up and then we can see what's going on. So this is my setup. So one of the early things that we started to do when I first started 4 years ago working on this project, and I must mention, and I'll mention it again at the end, that these ideas are not mine. These are the ideas of my supervisor, James Nye, and our collaborator, who's an information theory guy, Alana Orlitsky. And they existed as kind of like a loose collection of ideas. And I think recently we have come up with a more formal framework for them. And so we've been able to generate more specific predictions. But at the beginning, it was just kind of like, well, what can we do with this? And so we were just kind of walking around blindly trying to figure out what to do with it. So one of the things that we thought about doing was asking what happens if we reduce that stop signaling noise within the colony. So it's worth noting that almost nobody studies the stop signal. There are, I think, 4 or 5 labs in the world that do, and that's probably being optimistic. So not that much is really known about it. So we didn't know if we could actually do this. We didn't know if we could actually take out bees that were producing noisy stop signals and reduce the noise in the colony. So we spent a couple of months in the first summer sitting there with our little omnidirectional microphone, because the way that we identify stop signalers is to actually hear that little vibration, that ee sound that they make, but it has to be coupled with this headbutting behavior. If it's not, bees make all kinds of sounds all the time for a million reasons. So we actually have to see the stop signal happen in order to collect that bee and take her out. So we very carefully collected bees that seemed like they were making noisy stop signals. We numbered them, we kept them off to the side, and then we observed what happened to the colony dynamics after that. And each one of these experiments took 5 or 6 hours to run. Imagine this is UCSD, this is in the summertime, everyone's wearing full bee suits inside of an insulated trailer. It was over 100 degrees every day. So it gets a little hard to take. Nonetheless, so we did that, and then at the end of the experiment we would often we would let the numbered bees that we had taken out back into the colony and just kind of follow them. So a lot of this was exploratory observational stuff too. We didn't necessarily have specific hypotheses about what was going to happen. But we wanted to see what happened, or what those stop signalers did the next day when we looked at them. So is there evidence that they are consistent stop signalers? And at the end of our months of investigation and then analysis of our video data, nothing happened. So can you reduce the noise in the colony? No, at least not that way, and not in any straightforward way. And it turns out that it's probably not the case that there are bees that produce noisy stop signals. It's probably the case that everybody produces noisy stop signals sometime. So then we asked, well, maybe we can add noise instead. But in order to add noise, we had to develop a way to mimic the stop signal, essentially. So in order to do that, we had to understand some characteristics of the stop signal. The first and most important one is, does it actually stop waggle dancers from dancing? And yes, my supervisor had published a couple of papers on it, but the analyses were a little bit different. So what we did is some of these data were collected before I started on the project, but we have 4 or 5 years worth of data where we essentially follow returning foragers as they enter the colony with these little omnidirectional microphones, and we wait for them to either dance or not. And it turns out that only about 11% of returning foragers will dance, and the ones that dance, we would then note how many circuits they did, and then if they got a stop signal, and then what happened. Again, 4 or 5 years to collect 76 dances. Takes a long time. And that's also another thing, because you're following with omnidirectional microphones, 100 degrees in the trailer, you start falling asleep. But at the end of that all, we actually managed to have this big data set, and actually now we have more data than this even, and we're doing other things with it too. And we took every waggle dance within that data set, and we divided it into deciles essentially. And we said, was there a stop signal or not, or how many stop signals happened within that decile, and then we compiled them. Because of course this is a problem when you're trying to compare things of different lengths. We had dances that were 1 circuit and dances that were like 100 circuits. So how do you put that all together? That's one way, and of course we couldn't use the dances that just had 1 circuit, and I think we cut it off at 3 or 4 because even 2 is going to give you some strange results. And you can see that the vast majority, or not the vast majority, but there's a big cluster of stop signals right at the end, which does at least suggest that that is a causal thing, that's a causal relationship, that stop signals are actually leading to the cessation of the waggle dance. Actually, and just to back up for 1 second, when I showed you the video of the stop signal and that bee froze, that's really the effect, is freezing, and that's why it's called a stop signal, is because it produces this stop. It's not because it stops the waggle dance, although it might or it might not. It's this immediate stopping, and it turns out that any vibration played back to the substrate, to the comb that the bees are standing on, will cause this freezing behavior, which happens to be useful for us. So another thing that we wanted to do was we wanted to ask how are stop signals distributed within waggle dances? Because if we're going to try to emulate stop signaling, we have to know something like that. And then we, again, used our giant data set of stop signals within waggle dances, and we fit a negative exponential distribution to it, which suggests that the underlying process that generated this is Poisson, which isn't that surprising because lots of natural things follow those kinds of distributions. And so we've got our rate parameter, and the fit is pretty good. It's not super great. I should mention that, oh yeah, actually, so when I asked the question, are bees neurons? So we're doing something else with this data set to try to work that out. And I didn't mention that there are basically 2 possibilities for how stop signal information gets translated into dance cessation or not. One of them is that every time a bee gets a stop signal, there is some probability associated with her stopping, and it stays relatively constant. The other possibility is that bees are actually more like neurons, that bees are biological integrators, which means that every time you get a stop signal, provided that they happen close enough in time, then the probability that you're going to stop dancing actually increases. Right, so integrating evidence. And so we're actually currently using this information, so this data set of, I think we have 200 dances now. This data set to fit, we're actually fitting leaky integrator models to this, some work that I'm doing with my husband, who's a computational neuroscientist. So he's fitting these leaky integrator models to see if it actually fits the pattern of stop signaling, if there's evidence that they're actually integrating. Okay, so now that we kind of know something, and then my supervisor had done previous work measuring the fundamental frequencies and the harmonics and stuff, the properties of the actual signal. Found out that the signal is about a 340 hertz pulse, plus or minus about 40 hertz. And it's pulsed for about 150 to 250 milliseconds, so it's a fairly short pulse. So we knew those kinds of things. And remember I said before that any vibration played to the substrate will actually freeze the bees. I wish I had a video of this. I have a few, but I didn't put one on my drive. It's a neat thing. So on the surface of the comb, you can see bees kind of like moving and buzzing around. And then you vibrate the comb, and everyone stops. It's pretty creepy. So some earlier work was done in '86. This is a guy named Michelson, this black line here. And what he did was he looked at the frequency and the amplitude required when you vibrated the comb to produce the freezing response. And this is the curve that he made here, which is very pretty, because when we tried to replicate this, so Michelson did it on a Holcombe, which is a little bit, well, actually a lot, not particularly natural kind of stimulus. So it's never going to be the case that a stop signal is experienced by everybody on the comb. So we wanted to replicate this on individual bees, and that's what these way crazier lines are here. And it more or less replicated. It's quite a bit messier than Michelson's data. But basically, the reason we wanted to do this is we wanted to make sure that the artificial signals that we were developing would actually produce the effect that we wanted, which is the freezing response. Incidentally, those different colors of lines are just, we broke it down into drones, the male bees, and then nectar and pollen foragers, because we don't know, maybe they have different thresholds, but they don't really seem to. Okay. The next thing that we did was we actually created an artificial stop signal. So we took a pure 340 hertz pulse, and we tapered the ends, because natural sounds have tapered ends like this. And this one is 200 milliseconds long. And then we had to actually test it. So the first thing that we tried to do, so actually just to go back to my setup here. So this thing, this is called a Vibrational Exciter. It is the world's most expensive vibrator. It is $10,000, but it delivers a very, very precise vibration. And here you can see we've jammed it into the comb. We're doing full comb playbacks here. But initially we started out just by holding this thing in our hand, and you can't see it, but there's actually a metal rod that sticks out of one end. And we tried to mimic a stop signal that a bee would do. But of course, bees are very small, and this thing is heavy and big. So we tried for a while to do stop signals on individual bees by just gently poking them with this rod. But then I took some high-speed video of it, and it was always, you see bees doing this to each other, and it's just like, and stopping it. And hours when we were poking them, they would be like, it's difficult to control that. So that didn't really work. And we recorded some data, and it basically showed that the effect of just poking them with the thing was the same as if we had vibrated them. Like, it was really disruptive for them. So that was, several months of work, and it didn't work out. So then the next thing that we did, and this is what we did last year, was we tried these Holcombe playbacks. So even though it's not particularly ecologically valid, because again, it's never going to be the case that everyone in the colony experiences simultaneous stop signals, we decided that we would try, and so we did. And so we collected a couple of different kinds of data. So our comb, we did a 5-minute pre-test where we would scan through with our omnidirectional microphone, and we do what's called, essentially we take a survey of the colony behavior. And of course that method is a little bit limited because you can only focus on one little spot as you're moving through, and you have to make sure that you do your sine waves, so we do it in a sinusoidal pattern through the colony. You have to make sure that everyone that's doing that does it at the same speed, otherwise your sampling rates are going to be different, all of these problems. So anyway, we do a 5-minute pre-test period and then a 20-minute period where we're doing the playback or not to see what happens. We also did a condition where we took video of this rather than, so normally we do this as just audio, so we have someone using an audio recorder. They're mentioning when they encounter some kind of behavior, so you yell out. We have 5 behaviors that we pay attention to. I'm going to go through the details of all of them. We don't necessarily know what they all do. But you have someone yelling it out, and then you get a temporal distribution of behaviors and how they change. But again, the other option was using video analysis, where you can now see all the things that are happening all the time. But number one, it takes undergrads 1.5 million years to analyze that stuff. So to pick out every behavior that's happening on one comb of a honeybee colony is really long. The other thing is, remember that I said in the stop signal, you have to get the vibration and the behavior at the same time. Very, very, very difficult to do that from video. So right now we're limited technologically. Nonetheless, a couple of things came out of this analysis that were, yay, we finally have some kind of sort of result. These are some of the other behaviors that we pay attention to. 3 different kinds of signals. We kind of know what this one does, the tremble dance. We don't really know what the shaking signal does, and grooming is just grooming, so bees are just grooming themselves. And then this is waggle dancing, and this is simply the proportion of bees on our video that would freeze every time one of these artificial stop signals was played. And we see that, on the whole, everybody froze to some extent, and waggle dancers froze, which was really the whole goal, was to make waggle dancers freeze. So we know at least it produces freezing behavior. But more importantly, this graph shows, so the 5-minute pre-test, the average amount, or the average proportion of bees that are performing waggle dances, and for the 20-minute post-test phase. And we can see that there is actually suppression of waggle dancing in the playback condition. Yay, our stuff works, finally. But the problem is that I suspect that if we want to test my initial predictions back here, if we want to test something about adding noise to a colony, that if you're blasting the entire colony with stop signal, this is going to exceed the capacity of the colony to adapt to it, I think. I don't know for sure that that's the case. So this year, and people are still analyzing these data, so I don't really know what happened yet. This year, we found some kind of compromise that I think will solve our problems, I hope. We decided that we could target individual bees without poking them. So if we vibrate the comb right next to them with a small enough vibration that doesn't vibrate the entire comb, so it doesn't propagate everywhere, that's a much more realistic much more ecologically valid system to work in. And as I said, we're still working on analyzing those data, so I don't know if it worked, and I hope it did. Either way, we will be going with either the Holcombe Playback or the individual playback next year to actually test our updated prediction, because I had this brainwave about the colony size recently, and I don't know if it's going to be the case or not. But I think that it should be, that the larger colonies will have a higher degree of background noise in them. And they should then be less responsive to increased signaling or noise. So that means that we don't actually have to manipulate the amount of noise. We can actually just test them with a playback and see how resistant they are. So they manipulate themselves in a natural way, which is better, I think. And then we can either do the individual or the comb playbacks. Again, I don't know if we're going to see anything if we do the comb playbacks, but at least the individual, maybe. We'll see. Oh yeah, and then I wanted to talk about how, so that's manipulating noise, right? So if we blast them with playbacks, that's essentially adding noise. But we can increase the signal too. And this is actually work from my supervisor demonstrating that bees were responsive to predators at foraging locations. He developed this thing that he calls the robo-predator. But it's such a big word. It's mechanized caliber is what it is. So it's a little tweezer that delivers a very precise amount of force, and you can train bees to go forage at a feeder, and you can attack them with this little pincer, just like squeeze their legs a little bit, and they don't like it, and they come back to the colony and they're like, ah, stop! And that's what he calls the robo-predator. So we can in fact manipulate the amount of real signal getting to them. The problem with doing this though is that then they have to be trained to a feeder, and that is also a fairly unnatural foraging situation. I don't know, we'll see what happens. So where are we at? So my talk is not going to be an hour and a half long. I actually meant to mention that at the beginning because I want to talk to you all about this. I want to hear if you have anything to say. So yes, stop signals inhibit waggle dancing. Not a big surprise. Bees maybe integrate stop signals. We don't know. It's not catastrophic for this model if they do or if they don't. It just changes some of the details. But it is something that we would like to know. Stop signals are noisy, probably, because it could be the case that we just don't understand all of the things that bees are responding to. Maybe the noise is adaptive. Hopefully we'll be able to say something about that soon. We can't easily remove the noise from a colony, but we can hopefully increase the noise in a colony. And we can also increase signal to some extent. Actually, I do want to mention one other project that we're working, actually 2 other projects that we're working on that will facilitate some of this work. So right now I mentioned that analyzing the data that we generate is really time consuming. And really the only way that it can be done is by someone watching or someone listening to an audio track and transcribing, which of course is how people have done it forever. But it would be really, really helpful if we could do continuous online monitoring for large chunks of time and be able to compile all that data. But right now there is no system that exists to be able to do that. So I actually have a collaboration with a member of the computer science department, and we're working on developing software that will identify and quantify waggle dancing in real time. So that would be super cool if we could do that. But then the other problem is actually logistical. How do you actually film inside of a colony? Because if you leave a colony open, number one, it's not very nice for them. But you can change the lighting, you can change the insulation. That's not the biggest problem. The problem is if it's covered in a sheet of some kind, the bees are always going to hang out there. There's going to be occlusion happening all the time. So how do you actually get a picture of the whole dance floor? There was just a paper published where the people claimed to have been able to do it. So apparently, and we'll see. And then the other problem is like generating huge amounts of video data. And that's another thing that I'm working with my husband on. So my husband works in a neuromorphic engineering lab, and they work on like event-based cameras. So these are cameras based on how the retina functions. So rather than having frames of video where you capture everything that happens in a scene, the only time you capture anything is when something changes. And so you can save a huge amount of space by just paying attention to events when they happen and where they happen. But right now, the resolution on those things is fairly limited. But we also have a friend who works in Paris at the Vision Institute who has developed identification and tracking software for DVS cameras. So hopefully something will happen with respect to that. Okay, so I guess stay tuned, and maybe we'll get to test this year, and maybe I'll publish a paper. That would be exciting. And so I want to thank everybody here. This is my supervisor, Dr. James Nye, and this is Elana Orlitsky. These are the two guys who really came up with this idea to begin with, and forever grateful. And James taught me everything that I know about bees. When I started working in the bee lab 4 years ago, I didn't know anything about bees. My background is neuroscience and behavior, actually, in mammals mostly, but some arthropods, but not bees. And my husband and collaborator, Fred, who works with me on these projects. So we've just written some grants that hopefully will allow us to work on this more full-time. And of course, an army of graduate and undergraduate students who collect these data because it's really time-consuming. And my small funding agency in the school.
Plenty of time for questions.
Yep.
So I noticed that your new sub-prediction that you had produced was a prediction regarding the colony as a whole, but these larger colonies still want to be treated as or that you still want to treat these large colonies as single units of some sort. But is it possible that there's different noise distributions based on sub-communities within that? So for instance, are there bees that arrive at 6 a.m. or something like that and have different...
Absolutely possible. But like I mentioned before, because bees move around all the time, the network structure doesn't stay consistent. So even and there's no actual evidence that they really affiliate with each other on individual levels. They identify each other by cuticular hydrocarbons, so it's like colony level identification, not so much on individual. So whether or not individual subunits would affiliate to the degree where you would have stable changes in noise distribution, I don't know. But I suspect not, but maybe. Yeah.
So it's part of like a two-part question. The first part is sort of clarification. So the bees, with that noise, they stop all the time?
They always freeze,
Like no
or
matter
almost
what, no matter
always
what
freeze.
they're doing, even if they're not dancing, or-
Yeah, so yeah, if you have a colony of bees, regardless of what they're doing, there are some small exceptions to that.
Okay.
So sometimes if they're engaging in this tremble dancing behavior, which is a kind of thing that's related to waggle dancing, but it's not waggle dancing, and they're really, really into it, and you blast them with it, they don't stop. Yeah, it seems they're really motivated to continue, or whatever is going on.
Okay, so if that's like a constant, how would the noise be adapted then?
What do you mean?
If that's a constant behavior, that given that noise, right, and given that they don't know the difference between noise and signal, and then that the constant behavior is that they're always freezing or they're always stopping.
Oh, I see.
Like how would you elaborate, like saying that the noise is adapted?
Well, so the freezing is only important insofar as it relates to whether or not the waggle dance continues.
Mm-hmm.
So the freezing in some ways is probably decoupled from the waggle dancing. But the fact that everyone freezes when they experience a vibration does suggest that they can become habituated to it even if they're not waggle dancing. So whether or not that affects anything that they're doing themselves at that particular time, they become habituated to that actual stimulus and will start freezing less and less. And that's what we saw with our threshold experiments, that when we were testing the individual response thresholds, that as we retested the same bee over and over, she would just stop freezing. And she wasn't a waggle dancer, she was just a bee hanging out.
No.
Yeah.
I'm just kind of curious. the freezing that you see in the whole colony when you vibrate it, could that be another type of response, say, a colony that's being vibrated by wind and it gets fall or something like that? Is that maybe more of a response to protect themselves or something like that?
It could be. I suspect that it's more of that it's kind of like a listening response. So
Right.
they're communicating using vibrations. So if you stand still, then you're going to get whatever that signal is if it's important. That's my suspicion. I don't know. Yeah.
Is there a reason why, in order to study the adaptation rate, you can't, say, just administer this pulse at random points throughout the day over a period of time and then measure not the individual bees waggle dancing, but whether they are more likely to congregate at individual food sources?
Oh, yes. Logistically challenging, though.
Okay.
Yeah. So bees naturally foraging, their range is up to 6 kilometers in all directions from the colony. So you have to, there are ways to do it. So there are harmonic radar tracking devices that exist, but these are really expensive and hard to, so it's always like this problem. You can go the other direction and train them all to feeders, but that's a highly unnatural, constrained sort of circumstance, so, yeah.
But there's another component to Tim's suggestion, and that is that the habituation is kind of forming some sort of negative feedback that has a homeostatic effect, right?
Mhm.
So it sounds like you could potentially change the rate of the stop signal and see whether habituation results in a more or less constant rate of waggle dancing.
Yeah, so that's what I would like to do with the online monitoring stuff. So if we can, because it's not that challenging to administer the artificial stop signal, at least to the whole colony, if that's something that's effective, so that's,
Right.
that's a thing. So if that's not beyond the colony's ability to adapt to, it's not that hard to do it. Or we could imagine some device where there's just like, certain points where it could administer like local stop signals or something like that. The problem is like, how long does it take for them to actually adapt to that if it happens? And then monitoring them constantly to actually see that. I guess that's the issue. Like, you could let it play for a couple days and then come back and test them. Yeah, it's certainly feasible. Yeah, I guess that we could do something like that. Yeah. Or who has-
Actually, I think it was.
I was just curious to know how age might play a role, like either the age of the individuals or the age of the colony overall.
Well, bees don't live that long, so a forager will live typically about 40 days after she emerges. And bees do have some age-dependent task switching, so for the first 10 days or so, they don't actually even leave the colony. So whether or not age, and then age of the colony, the biggest consequence as far as I understand, as the colony gets older, it just gets bigger. And then at some point, if it gets big enough, it actually splits, and then part will go somewhere else, and then it gets smaller again. And as they get bigger, they tend to get grumpier. They tend to get more reactive to at least disturbances. So will it play a role? Maybe. I don't know. That probably doesn't answer your question. Yeah.
In terms of the idea of the different sizes of the colonies, to the extent that you know, is the proportion of stop signals like equivalent across small and large colonies?
That's an open question, so it's very hard to measure that.
Right.
So, as far as our subjective observations go, I don't think that proportionally there is any difference. That it seems like it's relatively consistent, but again, you would predict that at certain times of the year it's going to be different than at other times of the year. So, and again, just to measure the background level of noise in a colony is so hard to do anyway because of the behavioral stuff.
So I'm not 100% sure that I'm not asking the same question as Tim and David here. But I am curious about just, because one of your questions was, do these colonies habituate to the signal, right? Couldn't you zap the colony just all day and all night and see if eventually they just start moving as though they're a normal colony again?
Yeah,
Seems
that would be the thing to do, yeah. And that was, I think, his question.
And then a follow-up question. You've been calling the stop signal noisy signal of some sort, but it seems like it also is a tracking measure for the general threat in the area. So it's a way to compare foraging patterns to one another. If there's a general high level of noise, and anything below that is the place to go, and anything above that is the place not to go, right? So it seems like it's functioning in a way that's informative. It's providing information about your environment. But
Insofar as anyone pays attention to who's getting stop signals. But I think that the only one that's paying attention to that is the recipient of the stop signal. So I think that observers are not paying attention to who's getting a stop signal. It's just really the dancer. So I'm not sure if information is being shared to any other members of the colony that way. Does that make sense?
because it seems like it translates through the dancers, right? If the dancers are stopping, then people can react to that, and if the dancers continue going, then they can react to that.
They theoretically could. I think that it's unlikely that bees are that sensitive to it. I think that the only way that they're responding to it is just by there's no more dance there, so they're not being recruited to that area anymore.
Yeah.
Yeah. If that's what you mean, then
Yeah.
yeah.
Oh,
Yeah.
cool.
Did you have a question?
No, that was answered.
Yeah.
I was interested in how the bees are communicating these paths. So you mentioned that they give directions that are relative to the sun.
Mm-hmm.
I assume they're not just leaving once a day, which
No.
would be one way for them to use the sun. So if they're not, that means that they have to be giving like the directions on exactly where it is right now. So is it the case that they're getting a waggle dance and then they're going and finding it, and then on the way back they're giving different directions? I mean, it's depending on how far away.
Okay, so first of all, a foraging run doesn't typically take that long. It actually is really fast. So the turnaround time, at least when we train them to feeders, and even if the feeders are, a couple 100 meters away or whatever, it takes them like 10 seconds to fly there. So it's pretty fast. Yeah, obviously if they're foraging further away, then they're going to have to compensate. And then the question, so number one, we don't actually know what they're using to generate that waggle dance. So what's the information? Is it optic flow? Is it something else? We don't know. And there's actually a graduate student in my lab right now who's working on that to see if he can figure out what bees are using, landmarks or whatever. Sorry, what was the rest of your question?
Just about whether over time, I assume, like, just to give an even weirder example, or maybe more canonical actually, now that I think about it, if you give directions in the morning for something, and then like later throughout the day, I mean, maybe it doesn't change so much in each passing of the communication, but if they're going to that resource all the time.
The waggle dance will change over time, as the sun moves in the sky. I should mention too that the waggle dance isn't super precise. So there's a lot of error in there. And actually there was an interesting paper that was published where there's another instance where noise might be beneficial, showing that with each waggle run, there's about a 15-degree error on either side of the true direction. And that actually, as the resource gets closer to the hive, so as the waggle run gets shorter, that error decreases. Or sorry, it increases. As the resource is further away, the error decreases, and it kind of makes sense in a sort of exploitation you guys know what I'm talking about, the exploration trade-off, such that if the resource is close, then it kind of makes sense to have a lot of error in there, because then you might accidentally find another resource that's close by if you don't have to waste too much energy getting to it. But if a resource is really far away, and in particular if it's like the only thing, then you should have very precise directions on how to get there so that everyone doesn't waste a bunch of time flying around not doing anything. So the dances aren't super precise, and they do change. And bees coming back from a particular resource dance for that thing immediately. So there's not that much opportunity for the sun to move in that time. But as I mentioned, there is some evidence that they can somehow compensate for the movement of the sun to some degree. But that's also a fairly unnatural situation for them to have to wait. Yeah.
So correct me if I'm wrong, but I think I recently learned in a Netflix documentary that around this time of the year, most bee colonies in the U.S. are brought to the Central Valley to pollinate the almond trees, since almonds are in such high demand now. And that can be quite risky for beekeepers, because I guess and colonies disappear or collapse, or it's, I don't know what might happen. But this, at any rate, this seems to be like a huge perturbation to the colony system to move them from another state, another environment to the Central Valley.
Yeah.
And I guess this is less about noise, but about how foraging behavior might adapt under such extreme conditions. Is there research on that? Or?
It's an interesting question. So actually, we had this idea to use our playback system to more broadly mimic, for instance, the truck noise that bees would undergo when they're moving from one place to another. As far as I know, nobody's done any research on it because communication in honeybees is not the sexy topic. The sexy stuff is the pesticide research and the colony collapse disorder research. And yeah, a lot of bees come to the Central Valley to pollinate almonds, but certainly not the majority of them. There's lots of bees everywhere. Incidentally, if anyone thinks that honeybees are going extinct or not going extinct, they're fine. Honeybees are livestock. If they get sick or kind of like shitty, then we just bolster the levels a little bit. It's the wild ones that nobody is managing that we need to be concerned with. And also, we're not going to starve to death if all the pollinators die. It would be really sad and crappy, and our food would be way more boring. But a ton of the stuff that we eat is not pollinated by pollinators. So all of our staple crops, any of the grains, things like potatoes, those are not pollinated by pollinators. I'm not saying that it's not important. Yeah.
So you mentioned that when you vibrated the whole situation, that some percentage of them across all the classes of them stop. And I was just wondering how, you mentioned when you prodded them with the big tool that it was kind of ineffective because it was maybe overkill. But is there a way to gently tap them to see how often that single stimulus gets them to stop?
Well, that's what we're working on now. So instead of poking them, we're just vibrating gently the cone that is right next to them. So a lot of the signals that bees make, actually, they direct towards the substrate, not to other bees, because it carries through the substrate. And they use that. So bees actually hear with subgeniculate organs, which are these vibration detectors essentially below their knees. So actually it makes sense to send your signals through the substrate if you don't want to contact the other individual. Yeah, so we're working on some, it's not a perfect solution because of course it's still not the same thing as a stop signal, but it seems to be better. At least it's not super disruptive the way that poking them is.
This question isn't directly related to your research. It's more related to the kind of backstory you were talking about in the beginning. That Nobel Prize-winning study that discovered the patterns of the Waggle Dance. I'm just kind of curious, how exactly did he figure that out? Like what sorts of
what kinds of experiments did Von Frisch do?
Yeah, and like how did he extrapolate that particular pattern?
Essentially, he trained bees to particular feeders, and then he was able to decode backwards from like the location of that feeder what those two elements of the dance meant. So he knew that the feeder was 300 meters away. He suspected that maybe it had something to do with the sun in the sky. He noticed that these angles changed, and so then he could look at where the sun was relative to the colony, and again just decode those two elements, which is the length and the direction of the waggle run. Does that kind of make sense?
Yeah. So I'm just wondering like what sorts of possibilities do you have to iterate through before you settle on?
I actually don't know, to be honest. I'm not going to pretend that I do. I don't know. Does anyone know? No?
Magnetism, maybe? Cardinal directions?
What's that?
Like the magnetic field or something? Like birds?
Yeah, that's true, but I'm not sure that in the 1940s and 30s people were thinking about that stuff. Because I think that that's fairly recent, like at least 90s when people discovered that about birds. I think is there, yeah?
Is there any evidence that stop signals are delivered to non-dancing bees?
Yeah, we see that all the time, so that's one of the things. Like, maybe that's noise, but maybe it's just because the stop signal has other functions that aren't Waggle
Or
Dance
it
specific.
could regulate noise too, right? If it either promotes other bees to start giving stop signals, or if it habituates them, it could modulate the amount of noise.
Yeah, there are a number of possibilities. There's an interesting affiliation between one of the other behaviors that we look at, which is tremble dancing. That's that one that I said was kind of related to waggle dancing. So tremble dancing happens, again, in negative context. So we know that bees that are poisoned, for instance, or experience like really the predator bite thing, come back, and oftentimes they tremble dance. And also bees that are nectar foragers that are waiting for help unloading the nectar that they have stored in their bellies will tremble dance if nobody helps them fast enough. So it seems like it's, and oftentimes those ladies deliver stop signals too, just sort of, they're like trembling and they're going ee, ee. And so it seems to have some other functions rather than just noise and just to do with the waggle dance.
If you think that's actually a possibility, though, of regulating the noise, if you're looking for a neural analog, you might look at the locus coeruleus in the norepinephrine system, where you have these electrically coupled cells, so they're not exactly integrating, but they're similar, and they go between this state of high tonic noise and the state of low tonic noise, but high phasic signal. So I could imagine getting a signal across by that regulation of noise, as well as just having the noise amplify the signal, right?
Yeah, so actually my husband and I are currently writing a review of noise in biological systems, and that, of course, fairly recent discoveries of modulatory effects of noise in a lot of different neural systems. So yeah, we're definitely thinking about it.
I have just a quick question about honeybee biology, since I was curious, and maybe this will be interesting. So bees have to forage every day, regardless if it's sunny or not.
They don't.
Do they not?
No. If it's raining, bees are wusses, man.
Okay.
If it's raining, they're like, they don't go with that.
All right, fair enough. I guess, yeah, so that relates to my question. Like, I know, like some ants have photoreceptors that are sensitive to the polarization of light so that they can tell where the sun is regardless if it's cloudy or not. I wonder if bees have that too.
They might, and like ocelli are often implicated in those kinds of functions, so maybe, I'm not sure. I'm sure that someone knows. I'm not a honeybee physiologist. But yeah, they normally don't, and I mean that's kind of their whole deal, is they're able to forage and build up enough stores so that they can make it through winter. So there are days when it's not dire that they forage, at least if the colony is big.
Fair enough. All right, well let's thank our speaker.